Excited to come here, excited to share some of the research we are doing in Corpo in our research group on indoor microbial exposures with you. I'm gonna, like when I was asked to provide a title for the presentation a couple of months back, I was a bit careless with just writing down something, and I realized that I have a lot of ground to cover with fulfilling everything that I promised in there. But I'll try. So I'll be touching on several different projects that we're having currently going on, so you get an idea of what we are doing there, and I hope that we can have a good discussion later on and go deeper in specific aspects that might be of interest to you. Our group in Corpo is specifically and historically interested in the both beneficial and adverse effects of indoor microbial exposure. So if we think about the adverse effects, obviously the first thing coming to mind is the moldy homes, which is a big problem in Scandinavia, a big problem also here, I'm sure, and the related microbial exposure. We still don't really understand what's going on in terms of which exposure characteristics of moldy homes really link to health. So I'll go into some detail later on in some of these projects. On the other hand, we know that rich, diverse microbial exposure early in life is associated with beneficial health effects. So children growing up on farms where there's a lot of very rich microbial encounters are protected from asthma and allergy, or less prone to develop asthma and allergy later in life. And it's kind of like what this graph also tries to illustrate here now for asthma. We just did a chapter on environmental microbiota and asthma. So it kind of represents very well what we are interested in. So on the one hand, rich microbial exposure associated with farming environments, having siblings, having pets, high biodiversity in the living environment, and also the natural establishment of the conventional microbiota being associated with a more tolerogenic immune response, away from airway hyperresponsiveness, and away from developing asthma, so with asthma preventive potential. On the other hand, predisposing exposures such as respiratory pathogens and mold in buildings, occupational exposures, having the opposite effect towards inflammation, airway hyperresponsiveness, and the development of asthma. So this is sort of like the big picture that we are interested in. A lot of the good encounters that might be happening throughout life happen in early childhood, like even during pregnancy, last trimester of pregnancy, and maybe first half year of life. And we're trying to sort of separate what are the inhalation. We're focusing currently on inhalation exposures, but obviously there's thermal contact to microbes, and we try to sort of give a rough weight of the size of the encounters per day in terms of the microbial cells based on the literature that we have, like how much soil gets stuck on an infant's hand, how much house dust is ingested, breastfeeding, and so on. There is a lot of variability in this picture, but this is kind of like what we are overall very interested in. Much of the studies, or actually all of the studies that I'm going to present to you today, will include next generation sequencing data. So basically methodologies that allow us to have a very complete view of the bacterial and functional microbiota in house dust or in the sample that you're looking at. And that's really a technology that has revolutionized our understanding of health and disease. For example, the Human Microbiome project, now maybe a decade ago or so, these tools were taken and transferred into homes, into indoor research. And obviously there was a lot of hope, and there is still a lot of hope that applying these technologies to environmental microbial exposure and health research will really accelerate our understanding of what's really going on. So a lot of the data I'm showing you coming from the next generation sequencing data. And I'm not saying that this is the one method that we should all be using. It's one tool. It's one tool that allows us it's a non-targeted approach. So you see pretty much everything that's in there. qPCR, for example, really targets what you're after. So you only see what you're after. There is a bunch of limitations of using next generation sequencing, especially in exposure assessment, because it's not a quantitative method. But anyhow, I'll present some studies using this technology. And I always like to show this graph because it just really illustrates to me the potential of next generation sequencing. So what they did in this study was it's the 1,000 home study in the US where they collected dust samples indoors, but then also from the outdoor door trim from the entrance area. And they analyzed it for the functional microbiota using next generation sequencing and basically develop a predictive algorithm that links the home, the location of the home, or the functional composition of the outdoor trim dust with the location of the home. And what this graph here shows is that basically when sending in samples from somewhere in the US with a quite high probability, they'll be able to tell you within 250 kilometers where in the US you're living from. And to me, it's sort of, I'm thinking that if the methodology is able to do that, it should help us understand like what's a home where people get sick, where there is an abnormal microbial exposures, where a home that has a beneficial health effect. So this to me, sort of the big promise of next generation sequencing. I promised a baby robot, and this is the study that I'll present here, and all the credits really for this work go to Brendan Bohr, whom I met, I think it was in 2015, and he told me about that he had been planning. He's a particle person. Well, he's very active in many things. He's at Purdue University right now, but he told me about this idea that he'll get carpets from homes, and at that time he was working in Helsinki. So he said that he'll get carpets of homes into a chamber and do some chamber experiments because he wants to understand how particles are resuspended while a baby is crawling, like how this impacts on the infant exposure, because there was very little known. So basically the experiments that were done here were performed with this, I think it was 17 different carpets that he collected from homes, friends and family and colleagues in Helsinki. And he brought these carpets into an exposure chamber, or into a metal chamber that he was cleaning beforehand. And then he was running these experiments, and he engineered this little robotic baby, 4-kilogram robotic baby that does this modified belly crawl, and I'll show a video later on. Basically monitoring in real time the particle sizes, particle exposure of the infant. He did measurements with the optical particle sizer and the fluorescent biological aerosol particles. So those are both real-time size fractionated technologies. And when he talked to me, I said that I need a microbial sample. So he managed somehow to apply this IOM sampler connected to a pump on the head of the baby robot to sort of look into what really gets airborne in terms of the particles and the microbes. So what I was really interested in is because we're doing quite a bit of epidemiological studies, and so the sample materials that I typically get would be floor dust. So people collect floor dust at early life in their homes, and then we do microbial determinations from the floor dust, and we follow up the kids later in life and see how does the early life microbial environment impact on asthma, allergy outcomes later in life. And I've always been challenging the idea of using floor dust for this, because we really don't know how well it really represents inhalation exposure of an infant, and that was a unique chance. So basically what we did was to characterize the microbial content in the dust of these carpets, in the infant breathing zone, and then also in the chamber air, so in the bulk air of the chamber. That's pretty much what I've been telling you already here. So it's the experimental setup. Brendan then also added on some walking experience because he wanted to see how does the child's exposure change, obviously during the crawling, but then also if you have an adult walking by, and how does this impact in terms of the exposure in the chamber or in the bulk air. Now this is hopefully working. Just to illustrate you how that basically works, and we can have a long discussion about this, that how realistic this is, or how well this really represents a baby crawling. To me, with all the limitations that are there, it's maybe as close as it gets, unless you really mount up a baby with this kind of sampling equipment. And Brandon really, I can't even begin to express my gratitude and admiration how he just arranged everything for us to add on another sample and so on. So it was a very, very interesting project, and this is basically what you see what happens when the baby is crawling. This is now the particle mass distribution, number distribution, size resolved. So you have the background and the baby crawling, and you have this puff of particles in the infant breathing zone of the baby, then the decay period, and then again the same thing. This is basically what we're following. So one of the things that I was specifically interested in is how well does the microbial content in the floor dust correlate with the microbial content in the infant breathing zone, and we did some quantitative measurements for that, qPCR. And basically the line that you need to follow is here, where we have the concentrations or the loads in the floor dust versus the loads in the infant breathing zone. And what we find is that for some microbial groups, in this case the Penicillium aspergillus fungi, correlations were significant and fairly good, 0.78. For other groups, that was not the case, and it's of course interesting, intriguing. Also, the dust load in the carpet did not very well correlate with the particle mass concentration in the infant breathing zone. We can discuss long about what this really implies, but essentially you might have sand, soil in the carpet that never gets airborne, so that explains why the dust load, or the amount of dust in the carpet not necessarily correlates well with the particle concentrations in the air. And there are microbes that are attached to each other, to soil particles, to skin flakes, that don't become necessarily as well airborne into the infant breathing zone as other microbes might do. So that was an interesting finding, and also really, well, I'll get to this in a moment. Another thing we were interested in is how compares, because the other option to floor dust is to measure somewhere in the air of the home when we do the EPI studies to collect cell dust. So we wanted to see how do these two sample types in the infant breathing zone and the adult breathing zone correlate. And basically what we find is so this is the bulk air in the chamber, particle mass concentrations, and this is the infant breathing zone. So we find a 4.6-fold higher particle mass concentration in the infant breathing zone compared to the bulk air during the crawling. And what was interesting also here is that we see a between 9-fold and 21-fold increase of the microbial concentrations in the infant breathing zone compared to the adult breathing zone. So quite a large, much higher exposure in the infant breathing zone. And again, it's not uniform for all microbes. So for some, you have a much higher concentration in the infant breathing zone. For others, not so much. And this is essentially also the same thing, showing now microbiota in the carpet dust in the infant breathing zone air, the bulk air, and this is on a large phylogenetic, so big phylogenetic groups. Firmicutes, Actinobacteria, Proteobacteria, Bacteroides. So basically what you see is an increase in the relative abundance of the Proteobacteria when we go into air in this vertical gradient, and that goes with a decrease of the Firmicutes in the dust samples that are higher in the carpet dust compared to infant breathing zone and bulk air. And with the sequencing you can go deeper and really look what are the taxa in there. And these are some genera: Micrococcus, Staphylococcus, Corynebacterium. And what is interesting here is that you find things like the Staphylococci where you have quite high relative abundance in carpet dust, and it gets lower and lower the higher you go in the vertical gradient with the air. And then you find this for Staphylococcus corynebacterium, and these are microbes that we have on the human skin. So our hypothesis there being that, you know, this is shed from the skin in skin flakes. You get a lot of that in the floor dust. We know that, but that doesn't necessarily go very well airborne. On the other hand, we see microbes such as the Acinetobacter, that it's, for example, much higher in the infant breathing zone in terms of relative abundance compared to the carpet dust and some others. So you really have a variation in the vertical scale in terms of like how well the microbes in your carpet dust get airborne, how long they stay in the air. It has to do with the aerodynamic diameter of the cells, whether they are attached to each other and so on. So essentially, our conclusion from this study being that, and I'm just now wondering whether there is somewhere a timer, because I don't know. Right there. Okay, perfect. So we see that the infant activity creates this concentrated cloud, and I didn't go into so these are conclusions that we had also from the other paper with the optical particle sizes. So when the baby crawls, it creates this localized cloud of particles and microbes. And to my understanding, this would be a major source of microbial material for the infant. The carpet dust really is a relatively poor proxy for the microbial content inhaled by the infant, and specifically problematic, in my opinion, is that for some microbes it seems to work quite well, for others it doesn't. So if we go into going really, if we want to pin down specific microbial exposures and link them to health, you can go very wrong. It can work out, but you can go very wrong. And equally, the bulk air measurements typically underestimate the exposure and also compositionally differ from the infant breathing zone. So basically, conclusion from this is that if we really want to link infant inhalation exposure to health outcomes later in life, we need to do a bit better with the sampling that we are applying. Okay, so that was the baby robot from the title. So now I'm going to go to the endomicrobiota, and I heard that people here are also quite interested in the whole mold issue and endodampness mold, microbial exposures. What are the exposures that really play a major role in this phenomenon? So basically, what we are looking at is that we have a clear established association between dampness in mold observed in a building and health effects. So working, living in a damp home, in a damp office, in a damp workplace is associated with adverse respiratory health effects, including the development of asthma, exacerbation, and so on. But really, what's happening, like what are the causative agents and the mechanisms, we are still today not very, very clear about what's going on. And one of the reasons for this may be that the exposure situations in a damp building is extremely complex. So it involves microbial agents, including cell wall agent, metabolites, toxins, secondary metabolites, VOCs that are off-gassing from damp building materials, particles. So it's not. People have for a very long time tried to pin it down to one exposure agent that is linking to health effects, and I think it's clear that it's not going to be that. It's going to be a mixture. I've been lucky enough to work with Aino Neverleinen, who is the grand old lady in Scandinavia with the moisture damage research. She started off in the 90s and worked for 30 years or so on the issue of moisture damage and mold, and we're still communicating with each other. And now I'm sharing my frustration with her, and she tries to encourage me still. But she coined this. She sort of took the Tolstoy citation in Anna Karenina, where he says that all happy families resemble one another. Each unhappy family is unhappy in its own way. She applied this to buildings and said that every moisture damaged home is different. There are differences in the structures. There are differences in the microbes that are there in terms of the moisture damage that is there, in terms of how people react to the exposures and so on. So it's really, we're trying to look for commonalities in something that is a very diverse phenomenon. anyway, we're still trying, and basically for the next few minutes, I'm going to talk about a study, the HIT study that we carried out in 3 climatic regions. I'll get to there in a moment, but basically the million-dollar question for us being that, do microbial exposures in the context of moisture damage explain some of the association that we observe between moisture damage and adverse health? That was the question that we're trying to answer. What I'm showing you now is basically we're in the process. This is ongoing research still. We're trying to explore the characteristics of the indoor microbiota in European classrooms in different climatic regions and investigate the associations between microbiota and moisture damage and dampness. A lot of people that I have to acknowledge, maybe first and foremost Rachel Adams at UC Berkeley and CDBH in the US, who is running the analysis with us, and then all the collaborators that were involved in the HIT project. It was a big European effort. Also, I'll try to walk you shortly just to give you an idea of the design, because what we're looking at now in terms of the samples are from these 3 exposure assessments. But basically what we did was, again, in 3 European countries in parallel, carried out the same protocol. So we identified schools with and without moisture problems with questionnaires. We sent building inspectors there to confirm the moisture investigations and do the detailed inspections. Applied respiratory health questionnaires to all of those schools, then broke it down to a smaller sample of schools that we followed up longitudinally. 2 schools without, 2 reference schools, and then 4 schools with moisture damage. And then we did in-depth microbial and chemical assessments in these schools. Not so much chemicals, but we did do some measurements there as well. And then a longitudinal health assessment of the children. We did respiratory, we did lung function measurements. We did, of course, diaries. We did all kinds of questionnaires. We collected from the teachers serum samples and exhaled breath condensate and so on. So a very rich data set that we can utilize in this analysis. And what I'm talking about now are dust samples that we collected from these schools at 3 points in time during winter, spring, and again the next winter in these study schools. We used these electrostatic dust fall collectors that we placed on elevated surfaces between 1.5 and 2 meters. Basically the idea being that over a period, I think we collected dust there for 2 months. The dust settles onto these electrostatic dust fall collectors. It's contained somewhat because there's some electrostatic charges in there. And then you extract the particulate matter out of these wipes, do DNA extractions, and we did the qPCR analysis and also sequencing. Just for the numbers, so we had the Dutch and the Finnish schools we had complete. We did this in a late stage of the study, so that was running between 2008 to 13, and then we got some funding later on, and the technologies evolved, so we tried to go back to the samples. The Spanish samples somehow vanished in some freezers somewhere in Europe, so we can only talk about Netherlands and Finland here, but in total some 500 samples that we analyzed. And this is basically what we are looking at. Here is still Spain there, just to give you kind of like the scale from this, but as I said, Spain is not complete. These are different microbial markers in Spain, Netherlands, Finland for each of these markers. This is endotoxin measurements, glucan measured from the cell dust samples, and then different functional Penicillium aspergillus group, total fungi, and gram-positive, gram-negative bacteria. And basically the pattern that we see is that Finland being off the scale, like 10-fold, up to 100-fold, and even more lower levels in the Finnish classrooms compared to the Netherlands and Spain, which is interesting because what we are seeing in the health analysis is that the Finnish pupils are most prone to adverse respiratory health effects in connection with the moisture damage. And there's obviously explanations for this. So if you go into Finland, all of our schools will have mechanical ventilation. The kids take off the shoes outdoors, the classroom. There is no carpets, rugs, or anything like that. There is no stuffed furniture. So it's a very clean environment, let's say, and like the incoming air is mechanically filtered. obviously our winters well, it's the same as here. We have complete snow cover for, well, if we're lucky 5 months, and if we're not lucky a bit longer maybe. So the environmental conditions also very different, but then also the building maintenance and use is very different. So I think that's part of the explanation what we're seeing here. And now I'm going to the sequencing data. So this was quantitative data, and now we're shifting to sequencing data. And I just shortly need to maybe explain what you're looking at in this plot. So each point here is one classroom sample, one settled dust sample. And each point basically represents the total, in this case the bacterial, in this case the functional community in a sample. If 2 points are close to each other, it means that the microbial composition in these 2 samples resembles each other, is close to each other. So it's basically a map of dissimilarity between the microbial composition of individual samples. And what you see here is these 2 separate clusters. So Finnish and the Dutch samples almost completely separate into 2 separate clusters, meaning that they are very different in terms of the bacterial composition and also the functional composition. You can match this also. People are very interested in diversity, richness of microbiota, and we see that this separation goes together with a high diversity of the bacterial and functional microbiota in the Dutch compared to the Finnish samples. You might be wondering what this cluster over here is, and I think in the next slide this clears out. So this is now the differences between the seasons. So as I said, we have two winter measurements and then 1 spring measurement. Winter in the phase 2 is green, and then the purple are these are the winter measurements. This is the spring measurement, and basically what's popping up here is a dominance of some microbial taxa, Epicoccum and Mucosferella, which used to be, I think, Cladosporium herbarum earlier on. So outdoor-related taxa that really pop up in spring very clearly. Interesting here that the seasonal variation is more clear in the fungi in Netherlands. In Finland, it looks a little bit different. So we have a clearer picture, clearer separation of the bacteria compared to the fungi. Here it also goes like the way you would expect it to be, to have higher diversity during spring back to winter. So it's a very clear impact of the season. Now moisture temperature, which is what we wanted to look at, and first the qPCR data and the cell wall marker data. And this is something that, if we talk about moisture temperature, is quite, I would say that this is sort of like represents what many studies find, not all. But what you see is like somehow an increase in the overall microbial levels in moisture temperature compared to reference homes or schools. As I said, not all studies are confirmative about this, but I think this is pretty much what we see. There is an increase. We are not on totally different scales. And again, I'm showing here Netherlands and Finland separately, non-damaged versus damaged homes. So significantly elevated levels, not for all markers here. The total fungi are non-significant, and the difference are not very strong, but elevated microbial levels. When we go and look at the sequencing data for this, we see in the Netherlands here now, and this is again the richness, and I'll explain a bit more. So elevated richness, but it's not significant in moisture damage compared to non-damage. And there is a significant difference in the community similarity, but I will show you in a moment the plot, and you'll understand. But basically, what this means is that there is a significant difference in this in the microbial community composition, but the effect size is extremely small. It's a marginal effect that we're looking at here. Nothing compared to season or geography. Same thing here for Finland. Here we do have significantly elevated richness in the moisture damaged classrooms compared to non-damaged classrooms. And again, this community similarity, we see differences. And here this is the plot for the Netherlands. So there is a hinge of a separation if you stare at it long enough. For the bacterial community, it's a bit clearer for the fungi, but the effect is really not very strong. So basically the message here being that the moisture damage effect on the endomicrobiota is not as strong as you would maybe hope it to be or think it to be. And that actually what we saw in high T here, or what we're seeing in high T here, is in confirmation with many other studies that are not many. There are not so many studies that have used sequencing, but basically most of the studies see an increase of taxa richness with moisture damage, which makes sense. As you increase moisture, you have more water available for microbial proliferation. So it does make sense to some extent to have a richer microbiota there. But very subtle effects really on the endomicrobiome. I'm not sure how much I'm going to go into this. I might just tell this without trying to pin this down too much on the slides. But we did also a study in moisture damaged homes where we followed the homes during the moisture damage renovation. Because one of the things that we think makes the moisture damage research so challenging is that homes, residential homes, are so different, and it depends. If you have pets, it's going to impact your endomicrobiome. If you have carpets, it will. If you have kids, so there's a lot of moving parts. So one idea was that we try to fix these around variables as much as possible. Have a moisture damage home and follow it up during through interventions to sort of like factor out all these other moving parts. That's what we did here. We did tons of qPCRs, we analyzed secondary metabolites, and then also did sequencing. And basically it's the same story there, that we do see some shifts, but this is basically, again, such a plot looking at before and after intervention for bacteria and for fungi, like a hinge of a separation, but there is no clear separation. It doesn't change a lot when this home goes through the moisture damage remediation. And no clear effect for fungi. One thing we saw also here was that the taxa richness was reduced. So that was interesting as well. And the other interesting finding of that study was that so basically what we are trying to do right now also in the high T, and then what we did there is to sort of go deeper in these microbial communities and try to find out which individual taxa are associated with moisture damage and which ones are not. In this case, we were looking specifically at microbial taxa that were reduced in the relative abundance during the moisture damage intervention and try to pin those down. We see that they are clustering in this group of former order of Actinomycetales, and then that sort of like links again to knowledge that we have from cultivation study that actinomycetes, streptomycetes, these spore-forming microbes are associated with moisture damage. So I think that's a promising way to go to really go and identify individual taxa that indicate the moisture damage. Obviously, the ultimate goal then being using these moisture damage indicators and see whether there is any health relation going on. And this is stuff that we're doing right now in the high T. I don't have clear results yet, but so far I can say that we do identify moisture damage indicators in terms of the individual taxa. They are not the same in Netherlands and in Finland. And so far we haven't seen any strong associations with health, but that's like work ongoing. So but I think we will get there with the sequencing to say that, these are taxa that are indicative of, if you find these taxa, they are indicating moisture damage. But in how far they will explain health effects remains to be seen, really. There's one, well, maybe I say a word about this. There is one study out there that showed a really, really clear separation of the endomicrobiota in damaged versus non-damaged homes. And it was a study that was carried out in Colorado after the Colorado flood in 2013. And it looked at homes in a small geographic area of homes that were flooded and that were not flooded, and investigated what's the difference in the microbiota there. And there you see a difference. But to me, it sort of illustrates how massive the impact of moisture damage has to be so that we can really clearly separate this on a plot with the microbiota composition. All right, I talked already a little bit about this. moisture damage being one of the factors that shape indoor microbial communities, but if you compare this to season and other determinants, it's not very strong. Bacteria are important, and the indicated taxa approach is promising to my mind. One thing that we are doing right now, a study that we have going on, is for us, because we find it difficult to pick up moisture damage associated taxa from house dust. So what we are doing is we are investigating some 400 or 500 building materials, damaged and non-damaged building materials, with the sequencing to understand what does moisture damage look like in sequencing at the source where it really happens. Try to identify taxa that really associate with moisture damage materials, and then try to track them into the indoor home. So that's one thing that I hope will help us a little bit. An important paper being this study by Haggerty et al. That came out last year in Indoor Air, basically being the first metatranscriptomic study. So basically looking at gene expression and that was a model in a chamber, I think, with carpets where they increased the relative humidity in the close to the carpet surface, 0.5, 0.85, 1, and I'm not saying these are realistic conditions, but extreme conditions, obviously. But what she sees, what they reported was that with increasing humidity levels, the metabolic activity, and specifically metabolic activity in allergen, mycotoxin, and pathogenicity encoding genes, is increasing. And that's, of course, something when we are doing DNA sequencing studies, focusing on DNA, we lose it. We count cells. We don't see the per cell production of metabolites, which is something that seems to be relevant in moisture damage. Very intuitive, and we've basically probably known it before, but that's the first really detailed metatranscriptomic study that showed that phenomenon. Now, I'm already late in time, but I'll still very quickly, I'm just going to say what we found in that study without going into any depth. But basically the story goes, and this is from one of our birth cohorts where we have, well, it's two birth cohorts, one being half farmers, half non-farmers, and the other ones being just semi-urban or rural homes. But basically, we're having this paper in right now and hope to get it accepted very soon. It's a very nice story. But basically, the take-home message from all this is that so we know that farming protects from allergy and asthma later in life, and we think that the microbiota are associated with this. We know that the microbiota in farmers and non-farming homes are quite different. And basically, in that study, we tried to understand how things go. And basically, what the paper concludes is if you grow up on a farm, you're protected from asthma. Not protected. The probability to get asthma is lower. If you have, and we defined what is a farm home-like microbiome. If you have a farm home-like microbiome without living on a farm, you're still going to be having this asthma protective effect, which really links very closely to that the microbiota play a key role. And that's, of course, a game changer also if we think about this, that if you could transfer this farm home-like microbiome into urban homes, you could prevent potentially a lot of asthma cases, in theory. So that's basically motivated us to this, and that's the last thing in the title that I need to cover, the microbiome transfer project. Basically, the initial idea was that we take dust from these farming homes, transfer it into urban homes, and see can we, if we take the dust and put it on a carpet at the entrance area, can we change the indoor microbiome of urban homes? Because that obviously would allow us to sort of like, could be an approach to restore a rich environmental microbial encountered at early life. And we wanted to do this with the farmhouse dust. We screened the farmhouse dust. Of course, we found antibiotic resistance genes and stuff that shouldn't be there, or that is there, but maybe you don't want to put in anybody else's home. So the next best thing was that we took forest soil, applied this on carpets, put it into volunteers' homes, and looked how we changing the indoor microbiome in these homes. And it's sort of like one of those studies that you know, I would have never thought of going to a Finnish forest. It was a really cold day, and we were digging out some forest soil, processed this in the lab, basically applying this onto the carpets, impacting it on the carpet with such a plate impactor. And basically that was the setup. So we said that we want to place this carpet at the entrance area so people come in, walk over the carpet, and we want to see how this distributes inside the home. So we had samplings, settle dust sampling here directly next to the carpet, but then also in the living area. We had active air sampling here and here. And also in the living area, we looked at the carpets in the living area, basically, and we performed this intervention 3 times. So we had a baseline monitoring this home and the microbial content in this home with settle dust and air before the first intervention. So we placed the carpet there, followed up for 4 weeks, applied again soil on the carpet another 4 weeks, so 3 times. So we're basically looking at temporal changes, like how long is this effect going to last if we put this carpet? Is there an effect? How long is it going to take? How well does it distribute? And then we did all kinds of settle dust sampling and active air sampling, as I said already. And actually yesterday I looked, the samples are currently the sequencing, which we did a quick pilot to see what's going on. So this might be just a pilot effect, and we'll know more later this year how it really is going to look. But this is basically the taxa richness. What we see, this is at the reference home where we did not an intervention. What we see is that we seem to be able to increase the taxa richness in the settle dust quite close to the carpet. Well, quite substantially, from 300 different taxa to 500 different taxa. Obviously, it's not the same in the different homes. The homes were very different in terms of how many kids were the pets and so on, because we want to see that whether this effect is something that we really see constantly. But basically, seeing this increase in taxa richness, also summarized here, which is one of the things we wanted to see, basically. We also see that the carpet introduction makes the settle dust somewhat closer to the soil in terms of presence, absence of taxa. It does not seem to change the relative contribution of human source attributed taxa in the settle dust, but there's still lots of open questions, and well, I'm excited to go ahead with this project and see whether what will be the findings. But just to give you a short idea, and this is, I think, where I will stop. And thanks for your attention. Well, a lot of material. I thought it might be a bit too much, but I welcome all kind of questions, please, on this topic.